Thank you so much for the introduction. It's really an honor to have been invited here to the Allen Institute. Today, my presentation is going to be focused on visual approaches to make data more accessible, comprehensible, and useful. In the life sciences, I think visual representation can serve two primary purposes. I think one, to explore data while the scientific story is still unfolding, and the other is to explain scientific results. And I think these two ends of a continuum recapitulate a little bit about what happens in a lab where in early stage projects where the data started to come out. I think there's a need to quickly get an understanding of that data in order to refine experimental design and validate hypotheses. And then as that project matures and we have findings that we need to communicate to our peers and sort of galvanize the community around it, I think visual representation, of course, is also important to develop illustrations and figures for that communication. And both of these purposes, they serve different goals and they're aimed at different audiences. But I would say that a grounding in making design decisions based on how we see and vision and how we understand and cognition, I think is very important. I'll give you an example of those things in a minute. My road that led me to cross-country to Cambridge, working at this interface between art and science, in fact, started just down the street. I graduated from UPS, and for 3 years I worked as a research tech at the VA hospital up on Beacon Hill. So by day, I would run gels and PCRs, and then by night, I would be taking courses in scientific illustration from UW. Eventually, I went to Johns Hopkins for a PhD in immunology, where I discovered scientific and medical illustration. And this was my first assignment in school. And it's of a drawing of a human hip bone done in this age-old technique of carbon dust, where you take carbon powder and you apply it to paper with a brush. And the details are then added with intact carbon pencil and then erasing back to the white of the paper. And this drawing took about 40 hours to do, a full week. And at the very end, in order to fix the carbon to the paper, you take a mouth pipette and draw some ethanol, and then you spray it across the surface of this thing. My vision was just having this carbon just basically run down the piece of paper. So it was very scary and nightmarish. And as you can imagine, many of my colleagues, many of the medical illustrators today prefer Photoshop so that you have multiple undos and you can save stages. Medical illustrators do these types of drawings. This is an illustration of the circulation around the elbow so that blood flow can continue when you flex and extend your arm. But I wanted to tell you about one specialty of medical illustration, which is surgical illustration. And as a medical illustrator, you're trained to be in tune with the surgeon in order to develop illustrations that are used to teach and to learn surgery. So there's this concept of sort of a surgical moment where often a tense and pivotal moment in a surgery where relevant anatomies have been identified, prepared for subsequent steps. What you're seeing here is one of the steps in a Whipple surgery for the treatment of pancreatic cancer. And you can see that the duodenum has been closed, the pancreas has been bisected, the branches and the arteries and veins, the vessel's been tied off, and the neck of the bile duct's been exposed. And it's these types of illustrations that contains this rich set of information that as I mentioned, surgeons use to learn and teach surgery. I know that this type of images makes anybody squeamish, but if it does, you might want to check your emails for the next slide, which is a photograph of surgery. So one of the things I always get asked when I show anything related to medical illustration is why not just take a photo? And it would be much quicker and simpler. But as you can see, surgical fields are very, very small, and that's done on purpose, of course. And with photography, you sort of just get what you see and no more or no less. And you can really just capture an instant in time with a picture. But whereas in illustrations, you can sort of capture a moment in time. And you can see that surgical pattern in the upper right. That took multiple steps to do. but it illustrated very nicely in one step. But I think the real enduring quality of illustration really is the inherent selective ability that the artists have to decide what to show and how to show it. Of course, in any surgical field, there's lots of blood, lots of fat tissue, but that's not really the relevant information. And so going through that selective process, we're able to hone in on what's sort of essential. And at the Broad Institute, I think what I'm doing is probably not too unlike that, sort of wading through complex data and trying to tease out the essential and trying to amplify the signal over the noise. And having spoken to researchers here this morning, I think we have sort of this sort of common challenge. And one of those approach that we take to trying to make sense of data and to draw out those interesting aspects of data is really through data visualization. And Anscombe's Quartet, I think, is a really compelling example why we might want to visualize data. These 4 sets of numbers are statistically identical. They have the same mean, the same variance, they have the same linear regression on the x and y, and 16 other statistical metrics that we would put to these types of pairwise data. So while tables of numbers are highly precise, it's hard to tell how they are different. But if you were to plot those data, it really only takes a blink of the eye to see that they are wildly different. Anscombe is, of course, a statistician, and he wanted to point out the dire effect of outliers. And so these patterns are somewhat artificial, but they're not too unlike the patterns that we're looking for, overall trends and clusters and so forth. This is a simple scatter plot, so there were not many graphical choices that we had to make. But data, for the most part, has no natural form or no natural look, and so we have to decide how they are to be represented in order to make sense. By the way, I invite any questions along the way. It's not important for me to get through the whole slide deck. It's more important for me that you're engaged in the content and I answer your questions along the way. So please, if you have any questions. Okay.
How frequently do you use photographs yourself, for example, of those surgical? Are you present during the surgery and that's how you mostly derive the information, or you actually use multiple photographs to draw?
We do both. And so you study the case beforehand, and then there's always variance in the surgery. And you're standing over the surgeon's shoulder to make direct observations, but then you go back and you use reference material to do that work. So there are only really a handful of ways that you could represent numerical data. So for example, 3 pieces of data X, Y, Z, you could use shape to represent them. It could mean apple, oranges, and bananas, for example. If it's numerical data, you can use something like position on a common scale, or you can use position on a 2D scale, which is a scatter plot. Of course, you can use a length encoding. The longer the lines, the more of something, and that's the basis of a bar chart. But you can also think about length as width or height, and those are the basis for these types of diagrams called Sankey diagrams that we often like allows us to look at the provenance of data, how a data set or how a compound library evolves over time. And these are 2 published examples of Sankey plots. Of course, you can use color, different attributes of color. You can use the attributes of color that you can name, like hue. You can use saturation, the amount of that color, or the lightness, the black and white quality of that color. You can use area and volume. These get a little bit more complicated because you have to look at R square, R cubed for area and volume. You can use things that are more strange, like angle or slope. But here's an example from the New York Times that uses color, angle to tell a very rich story about how salaries have changed over a 30-year period and whether they're held by men or women. Registered nurses' jobs have really gone up over that 30-year period, and they're held predominantly by women, whereas construction foreman jobs have really gone down in that 30-year period, and they're predominantly held by men. So a very rich story using angle and color as a encoding. You can use things like curvature or arc length. along with angle, that's the basis for a pie chart, and to some extent, area as well. But you can also evolve that encoding into these sunburst plots that we prefer, which allows you to show proportion, but also the hierarchical aspects of the data and how they're related to each other. But that's basically it. That's the whole tool set that we have for visualization, and it's really a matter of how we use those things in combination. But not all those encodings are equally easy to read. For example, in an area encoding like this, how many times larger would you guess B is compared to A? I heard 20. 2? 10? The answer is 14. So it can sort of be challenging to do. So arc angle, for example, like if you were to order these wedges in order of descending, largest to smallest, what would you say that would be? Right. And so that same data, using length encoding, now from largest to smallest, that task is seemingly much easier to do with a length encoding, right? So some visual estimations are much more difficult to do than others. For example, if we want someone to see the difference between these two trends, it might be more effective to plot the delta between the two trends itself than to ask someone to do this visual estimation, because looking at this, you would say that the two trends seem to be converging, but in fact, the delta between x and y remains the same. So there's something that's going on with the visual phenomenon, us reading graphical information. Cleveland and McGill, while they're working at the Bell Labs, published this paper in Science where they looked at all those different visual encodings and see how fast and how accurate people are able to extract information encoded in these different ways. And they published this paper in Science in the 1980s and ranked the different encodings. And so here's a summary of that work, going from most accurate and efficient on the left with position to least accurate and efficient on the right with color intensity. And the last two columns doesn't really make sense for quantitative information. But so for when we have to invent new visualizations, these types of guidelines are very useful, and we might try to use encodings that are on the left side of this table. But of course, there are trade-offs. I think at times it's one set of encodings more intuitive than the other, while the other ones is more accurate. I'll give you an example. So this is a common way that distributions are represented with a histogram, where the taller the profile, the more of something. But if you talk to scientists about this visualizations, in this case, what they really were doing was squinting to try to see what was on the tail of this distribution. And in this case, these were cell lines that were dependent on a particular genes past the score of -1. So you could imagine trading in a encoding of height with an encoding of intensity. So instead of height, meaning higher quantity, in this case, the more intense the color, the more of something. Right? So this is using intensity as a proxy for height. So this gives you a sense of the center of mass, but also the individual data points. So we've done this in every case on the Cancer Dependency Map portal, where we have a distribution. We also show a raster plot. And the advantages in an interactive environment like this is you could grab one of these data points with your mouse and get some additional information, of course. They also tend to be much more compact. And you can see a pattern here that some biological processes are more like this, and other biological processes are more like that. The detail doesn't really matter. But this reference distribution has 161,000 items. And so if you were to plot that as a histogram, it would be off the page of the paper. And in publications like Science and so forth, where space is very precious, these types of representations can be very useful in trading out height for intensity. I think data visualization is really powerful because I think as humans, we're sort of walking pattern machine. Like we're always looking for patterns everywhere we go. And one of the ways that I think we look for patterns is that we group things that look alike, that are placed closer together, or connected by a line, or contained within the space. And we're always doing this search. But the interesting thing is that these visual cues get increasingly stronger, and I'll show you what I mean. If I show you these 4 objects, you might make 2 natural groups. There's a circle group and a square group. But if I move some of those objects closer together, that grouping seems to change now where proximity is stronger than likeness. So there's a group on top and a group on the bottom. And now if I connected some of those members by a line, that grouping changes again from top to bottom, and a group from the left and on the right. I think stronger still is containment, and I think the much more natural way to read this would be that there's a group inside the container and a group outside the container. So the strategy and the goal is to make design choices that are aligned with how we tend to see data and take advantage of these sort of visual phenomenon that's happening. And here's an example of a visualization that we developed for analysis, but eventually also used in publication called a sparkler plot. And I think without those connecting lines, which is stronger than proximity, you would just see clusters. But what's important here is the angle and the magnitude of those data points that tells this story that's related to looking at the lung cancer study. But that's the idea. And there are some visualizations that are more intuitive on others, and there are those that just stresses out so much that we can't perform those types of visual groupings and become somewhat not very effective. So this is an example from my home institute at the Broad. It's looking at a region of the genome, and we're trying to plot 8 different types of mutations at this locus. And some of those are different types of mutations that are keyed down here. And so one of the things that you're trying to perform is define all of the objects of a certain kind. So we can all try to look for frame shift insertions, which are downward pointing triangles. But I bet you by the time you find the 3rd or 4th one, you've already lost track of the first one. So it's really hard to try to assemble a pattern here to see what the distribution of the mutational profile looks like across this locus. And so I would say that this is sort of somewhat of an ineffective visualization. And to try to understand what makes something easy to see and something more difficult to see, we found it useful to sort of dig in a little bit into perception. And I think when we see, there's 2 processes going on. There's the bottom-up process where light is just falling in our retina and we see things. And there's a top-down process where we're looking for something specifically. And if you were to look for all the women in this picture of people at a soccer game, it takes a bit of work to do. But if I ask you to look for the man wearing a yellow jacket, it just jumps out immediately. And it's this sort of top-down process, I think, that has implications for designing visualization. We want to make what researchers want to see easy to see, the most relevant piece of information in a visualization, the easiest to be able to extract. Here's some more example here of amino acid sequence that this alanine is fairly easy to pick out. But the proline takes a bit of work because you have to do this sequential scanning. Of course, you really know what the P looks like. It just takes some work to do unless that P is also wearing a yellow jacket. That doesn't just happen with color, but lines that are placed at orthogonal angle. This is fairly easy to pick out. But a line at this angle with this number of distractor gets more difficult to do. So cognitive scientists describe this as a pre-attentive variable, so something that you can see in 250 milliseconds or less, regardless of the number of distractor. These things are very sort of special. And so I brought a series of animations that will play for about 250 milliseconds. And your job is to see if you can spot the object. So you're looking for that red dot in this field of distraction. I'll play a series of animations for 250 milliseconds, and your job is to say absent or present if you see it or not. Okay, ready? Okay, did everybody get that? Okay, so we're going to vary shape now. So that was color, and this is shape. And just to be fair, it's a little bit more difficult for people in the back because circles and squares tend to look more alike for the way you are. But they're also the default set of shapes. Usually in most visualization programs, we'll start with a square, then it'll go in a circle, or vice versa. So here we go. I heard some yeses, though. Great. So what if we vary both the color and the shape? And let's try this again. So you're also looking for the red circle in this case. Okay, here we go. You'll get a prize if you get one. Did somebody see that? So this is the problem with visual conjunction. So I think color is easy, shape maybe, but if you change both shape and color, it gets very challenging. But this is also the problem of the real world. We want lots of things to stand out at once, and this is what's going on here, is that it's changing color and shape all at the same time, and there's no way that we can assemble things based on proximity or likeness or connectedness to draw a pattern from this. So I mentioned these pre-attentive variables, and these are the types of things that are very special. Color, have you seen? You've seen there's size, orientation, shape, but it should be very different from another shape. You can add a mark to an existing shape. Motion, as you've seen, is very strong, which I think is very underused in visualization. I haven't figured out how to use that in our visualization, and you can use grouping. So the design lesson here is really to get things to stand out, you need to make them look very different from one another, and saving these pre-attentive variables for the most relevant aspect of the data set to make it most salient. I've mentioned color as a special variable, and it's one that we tend to reach for first often when we build visualization, and it's also very closely tied to what makes us think things look visually attractive. But I think color can be very challenging, and Edward Tufte said that color used poorly is worse than no color at all, and I sort of tend to agree with this sentiment. And I think the challenge is due to a couple problems. One is the problem due to the interaction of color, and the second is really just due to the hardware of our eyes. And I'm sure you've all seen these types of visual phenomenon where the inner two rectangles are in fact the same shade of gray, but they appear different to you. And even if I promise that they're the same, it's really hard to tell your brain to see them as the same color. But when you bring them together, you can, in fact, see that they are the same shade of gray. And this doesn't just happen in black and white. It also happens in color, so that the same color can appear to be different. And if you bring those colors together, you can see, in fact, they are the same color. And just as different colors can be, the same color can appear to be different, different colors can also appear to be the same. And so I think if you put this on different backgrounds, by all accounts, they look like they're the same color. So I think this, so color operates by this very deceptive logic, and they're very unstable. And so when we use color to represent quantitative information, I think we run the risk of messing with data integrity. And the heat maps, which is a very popular form of visualization used in the sciences, of course relies on putting a bunch of color tiles next to each other. And that same effect is present. So these two tiles that I've indicated here with the white dots are in fact the same color, but they appear to be different to us. And in one cluster at the top, you would say, well, it's nothing to do about it. But in the second cluster, it looks like a fairly high signal. But if you cut this figure and slide them together, they're in fact the same color. So it's a very popular form of visualization used in the life sciences is in fact taking the same color and making them appear different to us in some ways, sort of biasing the data. You might argue that, well, often we look at much larger heat maps, and we're looking for blobs of things, and we're not judging individual cells. But I think the effect is still there. It's hard to evaluate its total impact. And but this is one of the things we need to be aware of using color. The other thing I mentioned is some shortcomings about the hardware of our eyes. We have rods and cones to see different types of color, and we have cones to see color vision, but we have 6 times fewer cones to see light in the blue range. And so when we make rainbow color scales like this, while they make really fanciful pictures, it can break the discordance between the numerical scale and the qualitative color scale very quickly, where increment of 1 at this part of the scale, goes from green to seafoam green, but the same increment at a different part of the scale is more or less like a cliff. And so it can be fairly unreliable using a color scale. Even if we provide the color scale, it's hard for us to decode the information encoded in this way. So I think the safest way that we can deal with color is to really match the color key that we use for the data, and for quantitative information to use a sequential color scheme and only change the hue if it has a natural zero crossing. For colors that have order to them, to use color intensity, and then to use for categorical data, use different colors. So one way to pick color that are very different, as different from each other as possible, is to take advantage of all 3 attributes of color: hue, saturation, and brightness. And one way to do that is just to spiral out on the color wheel while you're changing the color intensity at the same time. And that way, you're relying on all sensors in the eye to help color discriminability. So I spent 3 years studying these sort of phenomenon from art and science, and read a series of articles published by Nature Methods. There are sort of 35 in total with my co-authors looking at these impact on visuals. Any comments or questions about this section? Mm-hmm. Anything that I found surprising in looking at color. There were actually more concerns about color blindness than I had anticipated. I've addressed that in one column, but that question keeps coming up, and which is a very important question about accessibility of data presentation. It's to pick colors that allow people with color deficiencies to read them accurately. And we found that by doing this sort of spiraling of the color wheel and using all aspects of color sensors in the eye, light sensors in the eye, that helps with issues of color vision. Yes?
With this spiraling view, I'm wondering if people, do they discern things that are less saturated and lighter as less important? And when you're trying to have everything read as equally sort of important but also be discernible, do you kind of run into that as an issue?
The questions were about the way these colors are picked and their sort of salience or their maybe contrast or attention-getting ability off of the page. And yes, that's the case, because you can't really have a dark yellow. Yellow it's not going to have a very wide dynamic range. And so you might want to assign colors that have a larger dynamic range and that's more saturated to get people's attention. Of course, there's also a cultural aspect about color. And so that red in one culture might signal something else and in a different culture. There's a question here too.
Yeah, so this is really great, important for us to know set of colors, predefined colors that are already validated by you, optimized by you, say this is the appropriate, set of colors that you should use to quantitatively reflect your data?
That's a good question. So she's asking about existing palettes of colors that might be useful. I provide one set, a colorblind set, but there are many tools out there that choose colors for you. And after the talk, I can give you those links if you're interested. Okay, last question's at the top.
Is there a convenient PDF bundle of your 35 articles available somewhere?
There is, and it's been put together. It's on the Scientific American store. I don't get any royalties from it. I'm just an academic who published them, but there is a bundle that you can get from Nature Methods. I brought one other vignettes about thinking about visualization and everything I've talked about and how to pack those together in support of a research project. And I thought I'd describe that to you. And it's part of this drug repurposing project where we've collaborated with a major pharmaceutical company to look at 400 drugs that's failed in phase 3 clinical trials, so for efficacy, not for safety. And in this project, we wanted to see how the compounds affected gene expression in a set of cells. Again, the full detail isn't so important. I just want to describe to you how we would take an approach to use visualization as an exploratory tool. But in any sort of approach of a project like that, we just want to start with a subset of data just to get started. So we started with 20 out of 400 compounds that's been identified by our collaborators as being high-value targets. We were using an assay that measures gene expression directly for 1,000 genes, and then would extrapolate that to the whole transcriptome. And of those 1,000 genes, we just focused on the 200 or so that have been known to be associated with disease. And we figured this is a drug repurposing project. This would be a good set of genes to start with. And so 250 genes, 20 compounds. If you were to plot that across 8 doses of the drugs, and so what you're looking here is a profile plot that on the x-axis are the 8 different concentrations of the drug. And what you're seeing on the y-axis is the gene level. So either the gene was elevated or it was suppressed across 12 different cell lines. Some of those are cancer cell lines, and some of those are primary normals in 2 different colors. And so that's what we did. And maybe something you would do as well, just take the data and plot it as you have it. That produced a 25-foot-long poster, and we took the first 8 or 9 feet or so, and we just put it on a wall. And then gathered oncologists, researchers around this poster and just asked them to circle whatever that looks interesting. And so we did that, and they noted some things. For example, this is a drug that causes this gene to be elevated only in 1 cell line, but not the rest. So that's interesting.
What does the dot mean?
Oh, that's a good question. She's asking what does the dot mean? It's our shorthand to say that that the level exceeded the floor or the ceiling of the plot. So we kept the plot consistent, but we just wanted to have a little indicator of that. And here's another interesting picture. This is a drug that causes this gene to be elevated in 1 cell line at a low dose and in a different cell line at a higher dose. So this is very interesting because dosing drugs is an important approach to this, and so forth. And but we realized quickly that this is an enormous search space, and this was just a subset of the data, and it would be very arduous to look through this space. And so we went back to the table to see, well, how else could we represent these data? And we noticed that the low-level data has a different amount of area under the curve than the high expressor or high depressor. And the transformation that we did was essentially took this plot and cinched the belt around it. So essentially just cinch its belt and transform this linear profile plot now to a radial plot. Okay, hopefully you're following me with me here. So this is the corresponding schematic of the plot. So that's the expression level at the third concentration in both plots. And that's the suppressed level of this gene at the second concentration for both plots. The concentrations go counterclockwise for negative values and clockwise for positive values. But you do that transformation. This is the same data plotted in these two ways. And you can see that it really quiets down the low-level chatter. And it really allows you to hone in on sort of the interesting parts of the data. And we think that these plots are more effective if you stand back further from that poster to see where the things are. We nicknamed these fly plots for their resemblance to fishing flies. And so in essence, we've created a new visual language. And so we started to learn how to read these instead of those plots. And so these sort of peacock tails, these colorful peacock tails, is perhaps an example of a differential expression. So that compound is causing the gene, a particular gene, in this case a PPAR gamma agonist, to go up in different cell lines at different concentrations. So that's what a peacock tail might mean. A fan shape off in one direction might mean something else. In this case, we have the mode of action of the compound, which is a lipase inhibitor, something that should reduce the production of fat, yet it's causing genes related to fat production to go up. So this perhaps is a picture of a feedback loop. We deliberately chose cool colors, blues and greens, for one types of cells, cancer cells, and then warm colors for normals. So if you see shapes that are predominantly cool colors, those are affecting specifically cancer cells and not normal cells. So, I don't know, sort of wacky of a plot. And I think it's really easy for us to look at these sort of things and then sort of dismiss them as being sort of not intuitive, like I don't get it, I'm sure no one's get it, and sort of dismiss them altogether. But I'm sort of here to make a bit of a plea. I think with that sort of attitude, we would really sort of limit ourselves to just a handful of plots. Some of those have existed for hundreds of years, and it not be enough for the complexity of data that we have to deal with today. This is the work of William Playfair, the Scottish political economist, who, among other things, have been credited to have invented the line chart in 786 and the pie chart in 1801. So at the time, most of the data he would have worked with was in tabulature form and tables. And so these graphic devices would have just been really odd. But of course, they've really become commonplace for us to read. And so I've been really curious about this area of study called perceptual learning. And if anybody here at the Allen Brain Institute does any of this work, I would be very interested in talking to you. But it describes the ability for us to learn a lot of complex visual cues that would allow us, for example, tell the difference between a trapezoid and a rhombus, and what a oak leaf look like versus a maple leaf. And so for some of us, a character for a house and family, or a positive sloping line or a negative sloping line. And so just as cardiologists are able to read EKGs and the subtle movements there, for pathologists to be able to read skin rashes, which to many of us look all the same, or pathologists to look at some set of complex visual cues and be able to identify patterns in it. I think we have the ability to do that ourselves and together sort of elevate the complexity of data that we can communicate to each other, so that by working together, people who produce visualization and people who rely on visualization to explore more sophisticated visual encodings, and then to learn how to read and decipher them, I think we can together sort of deal with more complex data. The fly plots, I don't think they're perfect. In fact, I think there are a lot of shortcomings. But we did this experiment by taking this to the Golub lab meeting. Todd Golub is the director of the cancer programs. There are about 30 oncologists there. Taught them how to read these plots and gave them a sheet each of a compound, one compound across 240 genes. And at first, people just sort of threw up their hands, but in the course of an hour, people came up with very interesting narratives of how these compounds might be repurposed. And some of those are worked up into repurposing hypotheses that we reported back to our collaborators. But I think visualization is not the full solution. And while they can be useful as an approach, I think we need a lot of different people to work together, people, user researchers, analysts who can really understand the problem that we're trying to solve, and technologists, software engineers, computer scientists who can make performance system, and think about the user experience so that we can develop environments where you can also have sort of a conversation with data, where you can ask a question, get an answer, and sort of follow your nose through the data. I think this is sort of the dream. And teams like this are not natural, I think, at the Broad, or perhaps not at the Allen either. But I think there are probably skill set distributed across the institute, but probably difficult to access. And we've been experimenting with new organizational structures where we are bringing those people together through a design affinity group that's grown to about 70 people who are embedded in different places around the institute to share those types of ideas I described. But what we realize is that really that's a peer-to-peer knowledge sharing group, and they actually can't do any work. And so we created a group called Pattern, which is a sort of a specialized team of designer and developer that can work through collaborative projects on approaches as the ones I've described. We've organized ourselves in the office of the chief scientific officer to make sure that we're aligned scientifically with where the institute is going. And so a lot of the work I've shown you today has been done by this team of Pattern. And I thank you so much for your attention. And I'm happy to take your question. Yes, in the back.
Here at the Allen Institute for Brain Science, we are quite interested in networks by which neurons or neuron types communicate with one another. So do you have any comments, recommendations, readings, or slides in your back pocket about effective ways of making graphs of network connectivity for different kinds of networks and so on?
You're asking about networks itself, where you have a node and a link and drawing these actual graphs. And we tend to refer to those as hairballs, and they're indecipherable hairballs. But there are mechanisms to display the graphs differently that would prioritize either on the node or the links. But I wonder if we put some effort into reading these hairballs, that we would actually get more out of them. So I think a lot of times the first reaction is that they're hard to interpret, and I would be interested in having a group committed to learning to read those visualization. So the answer is yes. I have resources about how to represent those types of encodings differently, but I also want to make a second plea about learning how to read those graphs.
Yeah, wonderful.
So one of the questions which you have not mentioned much is the fact that we can use computer vision and there are statistical analyses to be able to select patterns. You're trying to focus so much, especially when talking about perceptual learning, on elements of saliency, which we already understand quite well, in which how to pick a subset of data which is statistically different than that. Is there an aid for some level of computer vision which can come into play to be able to select, rather than trying to make use just of the human vision, to try to have more of a combined element in which there are statistical analyses to pre-select particular patterns to be able to display when you're looking for things?
I feel like this type of question almost hinges on the metaphysical. Because in some ways, we're going to visualize data and put it in a visual form and ask the computer to read that visual and tell us what it means. And in some ways, you make the argument that why not just compute on the data all the way through and get meaning that way? Why go through the visual aspect at all? There are certain visual estimations that have shown that are more difficult than others, and certainly I think computer vision could help. But I think you could just shortcut that path and just cut that part out of it, honestly. I don't think visualization is a solution to many problems. In many problems, I think the computation alone is sufficient.
What I was trying to refer to is there are good parts of everything. So human vision is fantastic. Collaborating with experimentalists, you can see fantastic patterns very rapidly out of the data, but very often you pick them up. It's very interesting that, for example, if you pick a human plus a computer as a chess player, they beat humans and they beat computers because there are strengths which can be enhanced. We don't necessarily use the same patterns, but some of them, some of these elements where you just need to pick something which is different, you might not need a visualization for that. You might be able to do it very rapidly with an algorithm. And there are some other elements where an algorithm is going to be terrible of discovering new patterns, which we don't have a good solution in intelligence, in artificial intelligence to do that. Why not try to aim to combine the two?
Yeah, I think I agree with you. I think that's the approach. Is keeping the human in the loop can be productive. And having the computer and the human support each other. I agree. There's a question up front here.
I had a question if you had thoughts or tricks on effectively using transparency and how to do overlays of colors correctly, given what you were talking about with the Alper's interaction of color work. And then kind of on a maybe related note, why with these fly plots you decided to take a linear thing and make it radial is kind of an interesting decision.
Okay, well, I'll answer the second question first, which is that on the transformation of the linear to the radial is that it becomes sort of independent on any sort of vertical or horizontal axis, so that when you have them in the matrix, it's you're reading them as a gestalt of the piece rather than making comparison. So if you have length or height, they might compare well vertically or horizontally, but not in both direction. And I think the radial axis helps with that. The second question about transparency, I'm not sure if I understand it fully, if there are ways to do that. I think some of that is a bit of an art and a science of sort of dialing up the amount of transparency so that things become salient. So in that example of the raster plot that I showed, we experimented with different levels of transparency so that when there's the center of mass would add up to something that's close to a solid. So you still have that dynamic range. Okay.
Hi. I'm curious to hear your opinion on UMAP and TSNE and that sort of these newly popular ways of visualizing high-dimensional data. It seems that from some perspective, they are similar to your fly plots, kind of a way of allowing a practice user to make new sorts of intuitive judgments of data set structure. But maybe from another perspective, it's more like the heat map issue where there are distortions that affect how we see it. Do you have any thought on the trade-offs there?
So the question is about UMAP and TSNE plots, which is a principal component reduction method, essentially. And I got away with not giving a talk without showing one, but you can't go to any talks these days without seeing a TSNE plot. And I don't have any thoughts about it. It's just a scatter plot in some ways, and we're really focusing on blobs of things based on proximity. But I have thought a lot recently about the discordance between how we display data and how a scientist would want the data. We have this temptation as computer scientists to display the data as we have it in the data structure and show it all. And a scientist coming to it, they see the world through very narrow lenses. They have a favorite set of genes, favorite set of cell lines or compounds, and they want to sort of get grounding on those elements first and then expand their world outwards. And so even when you show these TSNE plots, they want to know where their gene is or where their compound is on this map. And they see, oh, the neighbors are things I know about. Now I feel comfortable sort of going larger than that. So we've taken a different approach in the tools that we've designed so that it's grounded in very small elements of what a researcher would have a mindset of approaching a tool would say, I have a gene, tell me everything you know about this gene, and then make me feel comfortable and ground it there and then expand outward. So I'm not a big fan of those really large visualizations. I think they serve certain purposes.
I wonder what is the day job of your team?
Mm-hmm.
Do they develop new visualization tools? Do they work together with the researchers on the broad? The researchers just bring data to you guys to. What do you do?
Well, on sunny days like this, we would just skip work, but. So we work on a number of things. So one of the things we work on are large data-intensive portals. For this one is the Connectivity Map portal, which relies on gene expression as a readout for cellular states. You connect genes to drugs. We also work on the Dependency Map project, which is a large-scale profiling of cancer cell lines for genetic vulnerabilities and sensitivity to compounds. We also work on the GTEx portal, and so we're responsible for the visualization and the technical development of this whole portal. This is something maybe perhaps people at the Allen Institute have know about. We map out tissue for the whole body, but some of it's the brain as well. We also are involved in industry partnership. We have two currently that the team's involved in with IBM. One on cancer drug resistance, where we're developing a visualization tools to look at combination therapy and their impact on tumor fraction. We also have a collaboration with IBM Watson on cardiovascular disease and how to improve computes of the polygenic risk score as a predictor, and how to report that back to patients through clinics at the MGH, but also how Watson would do it for their work. We've also just launched last week a iOS app. It's our first for a clinical trial for IBD patients. And this was done with MIT and people at the MGH. We also try to empower scientists through hands-on workshops like these, where we teach the principles of visualizations and all of the things that I've presented here, and also give them sort of practical practice in developing their skills on the visualization. This was done recently in November for the Schmidt Science Fellow who came to visit the Broad. So that's sort of some examples of the things that we do.
So glad I asked the question.
Yeah, thank you. Thanks. So I had that slide deck, but I wanted to leave time for questions.
I'm wondering whether you have some suggestions on how to visualize 3D data, if you want to see through the volumes, what is the most effective way of doing it, and so how to explore this space more effectively? Yeah.
My opinion about 3D data is that if there's a natural spatial component to 3D data, and I think 3D data works. So a cell is 3D, a protein structure is 3D. But I think there's not a lot of compelling example I've seen to take 2D data and try to map in 3D. So if you have to look at three-dimensional data, it might be just as useful to look at them in pairwise in small vignettes, and then allow us to to bring those different views together to an understanding. So unless it's naturally 3D, then try to get away with 2Ds in multiple views. Okay, last question.
What if it's naturally multidimensional?
Like 50,000, that type of thing?
Genes,
Yeah.
We measure, 40,000 genes, and we're trying to get a relationship between these cell types, whatever.
Mm-hmm.
Yeah, multiple 2Ds, 3Ds look better.
Mm-hmm. I think we have a very hard time interpreting 3D plots unless they're moving, so that we can get some parallax and see that, and they don't print well in 3D in that way. I think that there are lots of techniques of using small multiples where you're looking at small subsets of a high-dimensional data set that have some purpose for them to be together. So maybe expression and vulnerability or something like that that would drive those pairwise comparison. Because I think beyond that, there's a challenge between encoding and decoding. And I showed one example from the locus of a gene there, where it's just because we can encode all the data doesn't mean that we can decode it. So I think we have to dial the visualization and cognition to that sort of sweet spot. Thank you so much for your attention and your wonderful questions.